All right, so thank you for the opportunity to speak today. Let's get started. So all of the networks that we study in connectomics are actually embedded in a meshwork of thin fibers. These fibers, or this meshwork or matrix, is present in all vertebrate clades. So you have it in mammals, obviously, you have it in birds, reptiles, amphibians, and fish. The middle panel is our own image of this matrix. These fibers, or again axons, have been individually labeled with different colors using the rainbow toolbox, and just shows how dense it actually is. So because it's present in all vertebrates, it is a fundamental feature of neural tissue. And this is a very important point, I think, because this has to be present in some shape or form for neural tissue to function normally. To assess the overall size of this matrix, I put in a circle. That circle represents a typical cell, the cell body, soma. And so some people have suggested that a single neuron is actually contacted by multiple fibers, and that's kind of obvious also just by looking at this picture. So if you make it a little bit more schematic, that's what we have. We have, say in this case on the right panel, a number of cells, but then we have overlaid these fibers, they're sort of overlaid on the system. So what are these fibers? You know them, but I'll be presenting a slightly different conceptual take on them. So these fibers release serotonin. They are unfasciculated in the sense that they do not form fiber bundles. They're very thin. Most of them are not myelinated. And the size, the diameter of a typical fiber is that of a bacterium. So what you can imagine essentially is you can imagine little E. coli swimming in your brain, your neural tissue, and producing these stochastically looking trajectories. That is exactly the point we're trying to make in this collaborative project. We're claiming that these fibers are strongly stochastic, and that makes them very different from the fibers, from the axons that many of us here study, where you can assume that A will connect to B in some kind of almost deterministic way. They release serotonin, but this is a bit of a misnomer in the sense that they can also release glutamate and many other neurotransmitters, GABA as well, neuropeptides, and so on. And so thinking of them as just serotonin-releasing fibers is a little bit misleading. They are very peculiar fibers. They do contain serotonin, but they may be more accurately described as serotonin-containing stochastic axons. What are they doing functionally? So serotonin, there's a saying in the field that serotonin is involved in everything and not responsible for anything. But now we seem to be converging to this notion that what this matrix does, what the signaling does, is fundamentally maintains neuroplasticity. So in other words, it keeps neural tissue in some kind of semi-solidified state. That is important in many brain states, wakefulness, sleep, and all of that, the action of psychedelics, depression, OCD, and so on. And very importantly, it also regenerates in the mammalian, in the adult mammalian brain. So that is unusual. If they get damaged, they can regrow quite robustly and recover. And they recover, of course, in new trajectories. So they don't repeat the previous trajectories. Again, emphasizing the point that that is an inherently stochastic entity. Now, this matrix, the serotonin, the density of that matrix, of course, has been mapped in all the gory detail in the brain. There are many papers that actually have done it. And this is one of the recent papers that sort of produced a beautiful map of these fibers. So what we're trying to do, we're trying to essentially understand how the stochasticity of individual fibers eventually leads to that stable matrix. So start with something that's inherently stochastic, and arrive at something that is sort of deterministic in terms of these densities. I'm not going to talk about other factors which are also contributing, maybe contributing to this. I'll also just show the slide that we are not just a computational lab. We do a lot of experimental work. These are two recent papers published by my two brilliant graduate students who are already doctors at this point. But this is just to note that we do study experimentally how these individual fibers behave. However, what I'll just jump to the actual models. So we're trying to model them as fractional Brownian motion paths. And I'll mention a little bit about what this stochastic process is, but essentially, again, they are stochastic, and the process is parametrized by a single parameter, which is known as H, or Hurst index. On the left, you see these fibers in the mouse primary somatosensory cortex. They've been stained for serotonin transporter. On the right, you see the model, the simulated trajectories. You see it now, there's a lot of similarity, including some bunching. If you look carefully, the little bunched points along the trajectory, and those get repeated as well. So this is 3D fractional Brownian motion. That's the way it would look. Actually, those of you who have worked with the serotonin trajectories, the serotonin fibers, actually would recognize that that looks similar to what these fibers are. In addition to that, we can capture serotonin varicosities in the same exact model. This has not been published yet, but it doesn't take much to actually model those varicosities as well. Top is Massimo Pasqualetti's lab, and that's an experimental mapping, and the bottom is actually the model result. So what is fractional Brownian motion? Very, very quickly, for those of you who are not familiar with it, this is a theoretical extension of Brownian motion. So Brownian motion, of course, is used to model simple diffusion. If you drop a little bit of ink in a glass, you get Brownian motion and normal diffusion. So it's an extension in the sense that it allows correlations among increments. And that is known more generally as anomalous diffusion. There are other anomalous diffusion models, but fractional Brownian motion is one of them. For mathematicians here, some properties are listed on the left. So if h is less than a half, you have what's known as anti-persistent motion, where trajectory moves back and forth. You might use it, for example, to model, using animal models of cocaine addiction, where it's back, forth, back, forth. They see that behavior. Also crowded environments and so on. Super diffusion is what we need. This is positively correlated trajectories, which then allow those paths to be maintained along a certain trajectory a little bit longer and more persistently than what you'd normally have with just normal Brownian motion. History of fractional Brownian motion is a gallery of brilliant people, as you can see, starting with Matthew Flinders, who circumnavigated Australia. Robert Brown, who is a botanist, happens to be on that ship, observes pollen motion, leads to Brownian motion. Einstein eventually makes it a mathematical model, and so on, and eventually we develop this process. Now, what we did in our lab, we actually extended even the theory of it in addition to doing actual experiments, in the sense that we study also what's known as reflected fractional Brownian motion. This is a complex mathematical problem, so we've been doing this hand in hand with physicists and applied mathematicians. And one of the properties of that fractional Brownian motion, reflected fractional Brownian motion, is that it accumulates at the borders. If you have the super diffusive, h over 1/2, it accumulates at the borders. And you have the opposite if you have subdiffusion. So we essentially showed that one can actually replicate a lot of distribution, a very significant portion of the distribution, actually experimentally observed distribution by using these fractional Brownian motion trajectories. This is one of the papers that we published, and so because my time is limited, on the right side, again, you see an actual, obviously, section through the midbrain, and the red one is just fractional Brownian motion in that shape. And now we just published a paper just literally a couple of months ago in a three-dimensional brain mouse model using supercomputing, because these are highly expensive computationally trajectories, and we had many of them. And this was done in collaboration with Thomas Voigt and Ralph Metzler. So it's it's a collaborative project with Germany. And we published a complete atlas of it. What would you expect, what the distributions would be if you simply had fractional Brownian motion behaving, kind of traveling through neural tissue in a realistic brain shape. So because I'm in my last minute, so we can see those comparisons in the published paper, we are observing an effect which I've kind of paid attention at for a long time, but people didn't, and that is that serotonin fibers tend to accumulate at the borders, and that doesn't seem to matter what that is. It could be a cortical layer 1. It can be, you can have a frog with its pallium and different architecture, you can have ependyma, you can have superior colliculus. They will accumulate at the surface. And so neuroanatomists may have misled themselves by paying too much attention to which neuroanatomical structure they're in. There's a very simple pattern which sort of does not care or what exact neuroanatomical structure this is. Now we are expanding it. The right panel is a little bit tongue-in-cheek. The left one, we will actually be studying this in sharks. I've done some work in sharks, so it's familiar. Different geometries, very interesting animals to work with. And my big point, yes, I think we have strongly deterministic axons in the brain, the ones that most of the people here study. But then we have a whole system of strongly stochastic axons, which again have, jokingly, we can call daxons and staxons. And actually, we can see this in the caudate putamen, for example. You can see actual bundles and which are marked with the red arrow, and then you see, these stochastic axons just weaving through them as, like bacteria. So this is just some publications we just had in a couple of last year, a few past, just in the past few years. And we also applying it, trying to introduce those systems to machine learning architectures, because those certainly are not present at this point. Right, so people don't even think about these kind of weird, moving trajectories in their, pretty much topologically organized machine learning networks. So we already published one paper, but looking for more collaborators. And these are the people who I just thank, my graduate students, and of course NSF, CRCNS, California Nanosystems Institute, and that's my brief presentation. So hopefully I'm not too much over. Yeah, thank you.
We have a minute for a quick question or two.
Do you think of other axons or systems that weigh this kind of?
Yes, of course. So you may be familiar with this concept of ascending reticular activating system, right? So ARS. And so, of course, dopamine axons might behave this way. We think it's a whole, it's the other half of the brain, essentially, I think. And we don't have a good conceptual take on this. I mean, we have some ideas, but that's a part that's been missing. Notice that in connectomics projects, serotonin is always left out. The assumption is they're kind of everywhere. They're not interesting. But there is, and all of us have unique trajectories, completely unique trajectories in the system, each individual.
Another very quick one as we switch.
Oh, you want to ask a question? No, so brief question. So when you compare the reality to the model, what are the metrics that you are using?
I'm sorry.
The metrics that you are using to compare the model with the reality.
Oh, so can you help me? Yeah, so at this point, because it's very difficult to study them dynamically, you need to isolate, we're using cell cultures and everything. But for now, we're basically losing at the resultant densities. So it's not perfect, but we're trying to get there with cell cultures and also some hydrogel systems, but it's challenging. But at this point, we're just do we get the densities that one would expect? Yeah, but it's a good question.
Thank you very much.